To convey my deep affection to this little animal, Drosophila, that seems to be recurring over and over during my past 20 years that I've spent studying living systems. So my first encounter was when I came here at Rockefeller to work with Eric Sija. He was working on regulation at the time, and so I didn't know much of biology. He was working on Drosophila, so that's my first encounter with this animal. And then I encountered again for olfaction, for flight. And then about 10 years ago, I started working with Stefano D'Italia on the early development of the fly, which is the story that I'll be telling today, in particular of the first couple of hours. So let's start from the beginning, which is the life cycle of Drosophila and the name. The name comes from Greek. It's a lover of dew, of humidity, because that's the kind of environment that the fly Drosophila likes to live in. And it goes that the female, you can distinguish females and males from, females are bigger than males. The abdomen that you see here is, this is the female here. The abdomen here, it's more pointed than the male. The colors are also slightly different. And the female lays eggs that you have here. And after about 24 hours, it depends on temperature, the eggs, they hatch, it goes into larvae, and then larvae go for a few days through cycles of mutations, it goes to chrysalis, and then finally you get the adult and the back to the cycle. What we'll be looking at today is this phase here of the first 2 hours, and there's a lot more, of course, to be said about the rest, but that's what we'll concentrate on. The fly has just a pair of wings and 3 appendages, the legs which are here. The second pair of wings got transformed into halters. Halters are very interesting for physicist objects because they vibrate, and vibrating, they keep the plane of vibration. So when the animal is turning, they act like a gyroscope that keeps the plane of vibration, and then it exerts a force onto the sensory apparatus, which is at the base of the halters, and that allows the Drosophila to make all the fancy things during flight that it's able to do. This animal is extremely a classical model organism in genetics, and the man who made it famous, and I cannot help showing this because it was done in New York, in Columbia, uptown. It's Thomas Morgan, who created, essentially took this fly, and he used it to, at the time, the big problem was that how the genetic information is transmitted. And he was able to show that the genes that at the time were a little bit fancy up, they knew that they were there. They were called factors. They were called elements. He could localize via some genetic screens and crossings that one of these genes, which was coding for the white eye on a male, was on the X chromosome, and this was the start of the genetics. And this quote is just to remind that the life of a scientist is not just a success. Most of the time, in fact, it's pretty frustrating because he was spending crossing pretty boring crosses of the flies without getting much for a number of years. And the fly room, the famous fly room, is shown in this picture here. You see the bananas because, as the name said, fruit fly, the fly loves the fruits. And you have all these bananas which we're making for the environment for growing the flies. And there's also an interesting movie, The Fly Room, which is, it's about 10 years ago, of the daughter of one of the assistant researchers in the lab, Calvin Bridges. So it's the story of this little young teenager. She was 10 or 11, not quite teenager yet, She was going to the lab together with her dad, so she was assisting to all the experiments of the father. She got on the field experience of the genetics going into the lab, and then you have all this flash forward of later life of this lady. And there's a very nice view of the lab, of the fly room at the time. Now, why is it important? In addition to curiosity, it's also because many of the Drosophila genes are conserved up to vertebrates and humans. The genome was sequenced in 2000, so it's completely annotated and sequenced. This is just the classical example of the Hox genes, which are coding for the Hox genes, and they are encountered in the segmentation of the abdomen and the body parts of the fly, and they're re-encountered in our vertebrae, in our back, and in our structure, as you can see here. And they're, of course, also responsible for some diseases and some bad mutations that you see in the hands over there. So there's interest also from the medical point of view, in addition to fundamental biology. As I was mentioning at the beginning, Drosophila is really a scientific gold mine. Olfaction, it's a fantastic system for studying olfaction. There's a few experts in the audience here. Flight control, I was mentioning the alters at the very beginning. It's also very important for vision, even though the visual system is not quite like ours, but they have a very good visual system, different than ours, because the functional needs are different. But it's very interesting to study and to get a sense of. So having said all this, let me now start with what we'll really be discussing today, which is the part on early development. Early development, the best way to introduce the problem of early development is maybe to show you a movie of what is going on in the embryo. And so I'm showing you a movie. This is the embryo of the fly. It's about 500 microns times 150 to 100 microns in the other direction. This is what will become the head of the fly in the adult. This is the tail, this is the ventral part, and this is the dorsal part. Okay? And what you see is you have the shell, and you're seeing the cortex. At the level of the cortex, what is happening. And it's sped up, of course, and you will see something which is of the order of the first 3 hours. So at fertilization, what happens is there's a calcium wave when the egg is getting fertilized. There's a calcium wave, and inside, as you will see later, the initial nucleus starts to divide. And you will see what happens at the cortex. So for a little while, you won't see much because the stuff is happening inside the embryo, and you won't see it because you see the cortex. And then at a certain point, you will see the nuclei that are popping up to the cortex, and then the action will start. So let me show you the movie. And right, so now the nuclei, they appear at the cortex, and as you see, they divide very rapidly. There's 13 cycles of division that happens in about a couple of hours. So these are phases that I'll discuss later of division. These are the waves that I'll discuss momentarily. And now we are at about 2 hours something. And this is the last phase between 2 and 3 hours, the 14th cycle. Here you have cellularization. As you will see, they start to create membrane. And then you have this phase, which is called gastrulation. Okay? Now, the movie, let me put a little bit more of a scheme of what is going on, because you don't see inside. So this is the beginning. It's the nucleus here. At fertilization, it starts to divide, and you see they stay inside. So this was the phase during which you were not seeing much at the cortex, because all the nuclei that were dividing were inside. And then at the cycles between 7 and 9, you have this ellipsoid of nuclei, which is shown here, that inflates a little bit like a balloon. And then the nuclei, they reach the cortex. And that's the phase where you've seen the nuclei emerging and reaching the cortex. Then during those phases here, you have the divisions that are happening. There's a few nuclei that are left inside that I will try to explain later what's the mechanism for this separation, for this first cell fate decision between the nuclei that reach the cortex and those that stay inside. And then at this stage, you have the division happening at the cortex with the waves that you've seen before. And this is the later phase that we won't be discussing today, which is the phase of cellularization, because the embryo at these initial stages is just made of nuclei. The nuclei don't have an envelope. They are not cells. They stay as nuclei, unseparated. This is the phase where you create the cells all around. And this is the phase of gastrulation that you've seen before. Now, one thing that for those of you who are used to the normal, quote-unquote, cells and the normal divisions, one thing which is pretty striking is the time scale of these divisions, because 13 cycles of divisions completed in 2 hours, you can calculate it's something of the order, one division happens in time scales of the order of a few minutes. So it's extremely rapid. And the reason and the functional need for this speed is very simple. The eggs are full of material which is laid down by the mother, and therefore it's very good food. And if they don't speed up, then do it sufficiently rapidly, they get eaten up, and therefore there's a clear functional and evolutionary push for completing all these phases as rapidly as possible. And therefore, what the nuclei and the cell do is that they manage to complete this cycle of divisions by suppressing the poses and the G phases that, as you see here, they appear very late in this. So you can imagine that during these phases here, there are just 2 things happening, which is the S phase, so the chromosomes are getting copied, without any poses, without anything. All the origins of replications are firing all together. There's very little transcription, which is the reason why the mother is laying everything into the egg at the beginning. So this way, the replication machinery and the transcription machinery don't get into conflict, and they can go the replication at full speed. And then you have mitosis. So essentially, it's a copying and separating machine that goes full speed in time scales of the order of a few minutes. And if we want to separate in different phases, which is good to schematize things, you have these initial 3 or 4 cycles where you have the initial nucleus which is dividing and creating this cloud of nuclei, which is of the order of 2 to the 4. During this phase, apparently there's nothing particularly interesting, at least we haven't found anything particularly interesting. Other than this division, they push onto each other by the spindles, and so they create this little cloud that you have here. Then at cycle 4 to 7, you have a first initial phenomenon, which is very interesting that I'll try and discuss, which is the axial expansion. So this cloud, which is localized here at about 40% from the anterior to the posterior axis, it starts to expand along the anterior-posterior axis. And you will see that this is due to very interesting hydrodynamic phenomenon that I will explain momentarily. Then you have this phase of the cortical migration, so the expansion of the balloon that reaches here. And then you have the divisions at the cortex and the waves that I'll be discussing later. And here we are at room temperature, we are at about a couple of hours. Okay? So with this scheme in mind, we can go back to the movie, and maybe I can show it again here. So here are the first cycles, at about up to 7. Okay, so you don't see for the reasons that I've mentioned before. Now we are about, right, so this is the cycle 9 that you've reached, you're dividing. Here they keep dividing, and here we are cycle 11 that starts to divide. Now you see these divisions that are synchronized that happen into here. Right? And Here you have the cycle 14, where it's posing. You have the cellularization, so you see that they separated a bit more. And then finally, now we are at about 3 hours, you have the gastrulation, which is creating this. Okay, okay, so let's start with the different phases. And let's start with the cycle 4 to 7, which is this phase here of the axial expansion along the anterior-posterior axis. So before we couldn't show things inside. In fact, there's the possibility of looking inside. And this is a good place. I start to show the experimental data. The experimental data, I don't do the experiment. It's Stefano d'Italia Duke, and this group who are doing these experiments. So everything that you see experiment is by his group. And what you see in this movie here is, again, the embryo, anterior-posterior, dorsoventral. But now you see something different, because what they have done is that they've put the red fluorescent protein on the so-called PCNA. Now, PCNA, you don't need to get all the details of what PCNA is, but it's essentially a clump that is associated with one of the DNA strands while it's replicating. So this protein is present in high copies during the phase of copying the DNA. So you will see that appears and disappears, appears and disappears, and the reason is that it's present just during replication. So during mitosis, you won't see any of this protein. It's just because the protein is not there, and it's present in S phase. But what I would like you to pay attention to is the fact that you can see the nuclei, and you will see the nuclei initially here that are spreading along the anterior-posterior axis, as you see in this here. Here the nuclei, as you see, they move here. So they span, they started here, and they cover of the order of 100 of microns in a few minutes. So that's the speed, which is pretty substantial, that you have to understand and to explain how it is getting generated. Now, tracking the nuclei is one thing. You can also track some particles inside the embryo. And so this way you can create the so-called Lagrangian trajectories. Lagrangian because you follow the particles inside the embryo. And this is the typical pattern of the Lagrangian trajectories that you see, and the flow that you have to understand how it's generated. So you can see there's 4 kind of vortices which are generated. You have a first vortex, which is here, where the particles, they move like this. Then they come down toward the center. Here you have a stagnation point where the velocity drops to 0, because you have the velocity which is coming down here, from down to up, then here and here. So here there's a stagnation point. And then you have a roughly a symmetrical structure that you have in the 4 parts here. It's not quite symmetric because the embryo is not exactly regular and it's not an ellipsoid. But this is the kind of patterns that is getting generated. And how this is getting generated is quite a non-trivial problem. Now, any fluid problem, the first thing you do is to estimate the Reynolds number. And the Reynolds number, it measures the ratio between the inertial forces to the dissipative forces. In this case, it's very small, which means that most of the dynamics is dominated by dissipation. And naively, you might think that because it's dominated by dissipation, everything is quite boring. And in fact, this is not the case. This is not the case. It's one of the lessons that you learn doing fluid dynamics in biological systems, is the fact that you can actually have very interesting fluid dynamics, even though the Reynolds number is relatively small. And very often this happens because there's an interesting coupling between the mechanical part and the fluid dynamic part and the biochemical part, which is so often the engine which is propelling the flow. So let me tell you at least a scheme of what is going on in this case. I will not get into the details of the equations. I'll be happy to discuss with those of you who are interested. So the first thing you typically do, because the Reynolds number is small, is you think that the the thing can be explained by the limit of the Navier-Stokes equation in the limit of low Reynolds number, which is the so-called Stokes equation. So you drop the acceleration term, and what is left is the diffusion, this is the viscosity, then this is the Laplacian, this is the velocity, and it's balanced by the gradient of pressure. So that's the limit of Navier-Stokes in the limit where dissipation is dominant. And now with this equation here, you can try and fit what happens into the embryo. So the Stokes equations are, since the time is completely off, and it's second order partial differential equation, it's completely dominated by the boundary conditions. So if you give me the boundary conditions close to the cortex, then you can calculate from this equation here, you can calculate what's going on inside. And so you can do this in this case of the embryo. And what you get are the arrows which are shown here. In blue is the experiment, in the other one is the data. So you more or less get the 4 vortices. You see some deviations, but overall, qualitatively at least, the overall scale is not too bad. It's the opposite. This is the red, this is the measured, and the other one is the Stokes. In fact, when you start looking in detail at how things really go, the Stokes approximation is not very satisfactory at all. And one way of seeing this is, for example, to calculate the vorticity. Now, the vorticity is the curl of the velocity. So if you take the curl of the velocity in this equation, what you end up is the Laplacian of the curl of v, which is omega, equal to the curl of grad of p. The curl of grad is equal to 0. So you end up with an equation that the Laplacian of omega is equal to 0. Now, Laplacian of omega equal to 0, it means that omega is a harmonic function. And the harmonic function, if one goes back to mathematical physics, harmonic function cannot have maxima and minima inside the domain. They must have maxima and minima at the border of the domain. Which means that in the case of the embryo, there shouldn't be maxima and minima inside the embryo. And when you go and measure the vorticity, you find clearly signs of maxima and minima and extrema inside the embryo, which means that considering this as only a Stokes flow doesn't work. Another reason why it doesn't work is that a fluid, the embryo, the cortex here doesn't move. It's fixed. Therefore, a normal Stokes fluid at the boundary like this, which doesn't move, should satisfy no slip boundary condition, which means that the fluid should actually not move close to the cortex. And therefore, it means that it's not quite consistent hydrodynamically to think that the fluid is moving close to the cortex here. So it's clearly there's something else which is going on. And something else which is going on without getting into the equations, but just to give you a sense of what is going on, it is important to consider the fact that in the embryo, there are different components and there are different elements that compose the inside of the embryo. There are components that are the soul. So the soul is almost like a liquid. But then there are other components that are more gel-like, and they're active. This is the actomyosin. And the myosin is, for example, the motors that are responsible for the contraction of your muscles when you move your muscles. And these two fluids, in fact, have to be treated separately. This is the conclusion of this recent work that we've done. And in particular, what the message that I would like to convey here is the following one. It's a scheme of how things are happening in the case of the Drosophila embryo. So you have the nuclei that you've seen before, which is localized around here. Now, the nuclei, what they do is that they secrete a component, which is called PP1. PP1 is one of the regulators of the cell cycle. And there's secretion and there's degradation. So you create a cloud of PP1, which you can consider as a sphere. Roughly, it's a sphere of about 50 microns of PP1, which is centered around the location of the nuclei. So you see this here. Now, this PP1 regulates the activity of the myosin via some intermediate molecules, which is not so important. So the myosin is responsible for contractions, and it's the gel part of the fluid. And contraction, it means that at this location here, where there's a higher concentration of PP1, the activity of the myosin is stronger. So there's a contraction which is going on at this location here that corresponds to the nuclei. Now, a contraction of the gel, what it does, because there's friction and entrainment between the gel and the sol, is going to take the sol and get the sol entrained at this location here that corresponds to the contraction. And therefore, the sol is doing this. It comes from this side. The sol is incompressible, so they come from the two sides here. They cannot go in this direction because there's a boundary which is fixed. So it gets inside here. It gets inside here from this other side. Again, it's incompressible at this location, so it must go to the side. And this is how you get this anterior-posterior flow that you've seen before, and how you get the 4 vortices that you've seen before in this structure. Now, you can put equations onto this. You can reproduce the whole structure. You can make predictions. So make a long story short, you can essentially understand the dynamics of this system as a good example of a coupling between chemistry and fluid dynamics into this. And this is just one of the examples that come up. There's some other examples that you can get. This is a late cycle. Even earlier than this, you can have in the ovocytes, you can have some flows, which are very important because they redistribute the material. So there's work which has been done recently across the street. And in general, there's a lot of activity on what is called cytoplasmic flows. Cytoplasmic flows in a broad sense. They used to, this is a review which was written a few years ago by Ray Goldstein. And this is an example of plants, how the plants, they have a big problem to transport stuff from over centimeters. So it's an even harder problem because they have to transport over tens of centimeters. So there's an issue of understanding how the stuff is getting transported over such large distances, which is the problem of cytoplasmic flows. Okay, so this was the first phase. Now there's a second phase, which is the inflation of the balloon, which goes from cycle 7 to cycle 9. Okay. Now, the molecular players were well studied and identified a few years back. This is a classical paper in Journal of Cell Biology. And the previous phase, it was actomyosin. In this phase, the major players are microtubules. Microtubules, with respect to the actin, are much more rigid. So you should imagine that microtubules are like stiff elements, a little bit like tubes that you have, that sprout out of the nuclei that you have here. They radiate from the nuclei. And then at the point of intersection between two microtubules coming from different nuclei, you can have motors that move along this nuclei. They exert forces. And therefore, via the interaction of these microtubules, the nuclei can exert forces onto each other, push onto each other, and regulate the respective position of the different nuclei. So the idea that was qualitatively sketched out in this paper here is that they did, first of all, genetic experiments showing that the microtubules are the essential element, because if you take them out, there's no migration and there's no inflation of the balloon. And the second thing is this qualitative scheme here that more or less explains how you can inflate by these forces which are getting exerted by the different nuclei onto each other. Now, and this scheme it's essentially right, okay? It's essentially right, but there's a few points that are important to explain, both from a quantitative point of view and about some interesting phenomenon, which is the following one. If you go, as I told you at the beginning, there are nuclei that reach the cortex, and there's a bunch of nuclei that, in fact, they remain inside. They're called the yolk nuclei, and they remain inside. if you go and measure how many of them they remain inside, there's about 6,000 that reach the cortex. The total should be 2 to the 13, which is the number of cycles that you have. So the fraction of nuclei that reach the cortex is about 73%. So there's about 27% of nuclei that remain inside. And you can track the nuclei. It's this bunch of red nuclei that get here. This is the time, and this is the distance to the cortex. So this is the 73% that reach the cortex, and these are those that remain inside. And it's unclear what is deciding which ones they go out and which one they remain inside. And it's not just a curiosity. It's not just a Kabbalistic curiosity of saying 73, 27, and so on. Because first of all, it's the first cell-fate decision. And second of all, the ratio between nuclei and cytoplasm, it's a very important determinant of development. In particular, it's being shown, for example, in this paper here by the group of Amodeo, that the nuclear to cytoplasmic ratio regulates transcription. And if you start messing up on this ratio, on this number here, there are effects and there are misdevelopment that get produced. So development doesn't proceed in the normal way. So what we've done, and I'll show you later who are the person who have done the experiment, is that we followed the nuclei inside the embryo that are here. I can show you what they look like. Okay, so this we are at cycle 7, and we follow them as they divide and they expand. And I'll explain momentarily what the colors are. So what the colors are is the following thing. When the nuclei, they divide, there's a spindle that is created that pushes onto the two nuclei and pushes them to a certain distance. And you can measure the plane of the spindle which separates these two nuclei, and measure the angle that the spindle forms with respect to the cortex. Okay? So if you have 0 degrees, it means essentially that it's parallel to the cortex. 90 degrees, it means that the two nuclei are dividing like this. And you can make a histogram of the angle of division at cycle, this is cycle 8, and this is cycle 9. And you can put the threshold at 45 degrees and count those that are parallel and those that are perpendicular, schematizing. And what these numbers here show is that essentially by taking perpendicular or parallel like this, you can reproduce the numbers of those that get to the cortex and those that stay inside. So the colors here is this division. The yellow ones are those parallel, and the red ones are the perpendicular. Okay, so the spindle is the major determinant. And now we can try and rationalize how the plane of this spindle, how it's getting determined. And in particular, what you can do is that you can create a tessellation. You have the nuclei here. You create a Voronoi tessellation of the ellipsoid, and you count how many. So you create a polygon around each nucleus, and you can count how many neighbors each nucleus has. On average, it's going to be 6, but in some cases, you're going to have 5 or you're going to have 7. And a fancy name for this is topological charge. If you're not into this, you can just count this, consider topological charge as the number of neighbors that you have. And the interesting remark which is conveyed by this plot here is that the spindles that divide in the perpendicular or in the parallel direction is very much related to the topological charge, so to the number of neighbors that you have. You see that when you have less neighbors, then you tend to go perpendicular, and if you have more neighbors, you tend to go parallel. So the number of neighbors is what controls the plane of division. And this is interesting in the field because the number of neighbors doesn't have anything to do with the distance. So there's a distinction which is made between metric and topology. Topology doesn't know about the distance, it knows about the topology, so the number of neighbors. So what comes out of this is that it seems like the topology of this tessellation here is what is important. What doesn't matter the distance, the size, but it's more the topology which matters. And one can rationalize this by the following simple argument. So you have two nuclei, which are here. They radiate the bunch of microtubules, which are represented here, that have a certain overlap in this intermediate region that you have here, which is represented like here. So now you can ask, what is the torque which is getting generated by these microtubules that get in touch with each other? And the torque which is getting generated is given by the density of microtubules that you have here, times the force which is exerted, and times the radius that you have between the two. So there's a factor R which comes from here, and then you have the density of microtubules, which of course goes like 1 over R squared because you have a cone which is coming out. And then there's another factor R, which is related to the integration all along the polygon that you have here. So you have 1 over R squared, you have R from here, you have another R which is coming from the total integral. So when you calculate the amount of torque which is getting produced from a mechanism like this, you essentially get something which is independent of R, which is a way of rationalizing why the metric elements don't matter much, and this is what the topology is what matters most. There's a way of testing this by doing mutants, where you change the distance of this, so you change the typical size of this tessellation. And indeed what you see is that the distribution of parallel and perpendicular doesn't change, which is at least an indirect confirmation of these arguments that we are making here. Okay, I have 5 minutes. So let me go to the last stage, which is the stage of from 10 to 13 of the waves. And again, let me show you a movie of the divisions and what we try to understand. So these are already late at the cortex, and now you will see around 30 minutes here, you will see waves that start from the 2 poles and they move inside here. Here it is. So you see that it starts from here, they start to divide, and then there's a wave of divisions that gets into here. And even more clearly, it's going to be around 50-something. Right, now it starts, and it moves toward the center. Okay? So the divisions don't happen like salt and pepper. They happen in a coordinated way, moving from they tend to start from the poles and they tend to move inside. And how is this wave created and what is the mechanism for this? It's the question and it's the problem that one can try and address. Now, of course, waves are not new. It's not an unknown phenomenon. And in particular, in a very simple modeling in physics, it's supercooled liquids. And supercooled liquids is the typical model. It's a reaction diffusion system with a bistable potential, like the one which is shown here. So a potential where you have a minimum which is here, a minimum which is here. And in the case of supercooled liquid, so liquids which are cooled below the 0 temperature very slowly, typically this would be the water and this would be ice. Therefore, the typical, the preferred, energetically preferred state is the ice. But you can get stuck into the metastable state, which is water on this side. and you need some perturbing factor in order for you to jump from water to ice. Typically in physics, the model is done, it's done adiabatically. You cool very slowly your system, and therefore, that's how you manage to keep your system into this, in this metastable minimum that you have corresponding to this. Now, this kind of mechanism, so based on slowly changing potential, is what was initially proposed as the mechanism of the waves in this paper here. And this kind of waves are called trigger waves because they get initiated they get triggered by the jump of your system from the metastable state to the stable state. Now, I told you at the very beginning, though, that Drosophila is in a hurry. Drosophila is in a hurry, and the cycles, they get completed over timescales of the order of a few minutes. So when you start thinking and measuring, putting numbers on this, you realize that, in fact, one of the main hypotheses and assumptions that you typically do in physics, which is that the potential is essentially static and all this process is very slow, in fact, it doesn't hold. It doesn't hold, and the whole process is much faster. And therefore, what we had to do was to rethink about this process. There's still a bistable potential, but there's something else which goes on, which is that the potential, rather than being static, is rapidly changing in time. And when you do this, without getting into the details, you have a potential like this, which is bistable, but this potential goes up in time because of the cyclins which are getting produced. And then the main mechanism becomes this saddle node bifurcation where you lose one of the minima, which is here. And what we discovered is that rather than having trigger waves, you have phase waves. Now, phase waves and trigger waves, you can think of at the mechanism of these two possibilities on a runaway of an airplane. So imagine you have all these lights which are here. A trigger wave would be that the first light goes on, and then there's a communication between the first light and the second light. There's a cable of electricity that when this goes on, sends a message to the second one, and the second one goes on, and then this one goes up again, and so on. So there's a coupling between the two lights, and there's a signal that must be propagated from the first to the second. This is the trigger waves. There's another possibility, which is that you have someone that regulates the time that all these lights go on. Okay? So this one goes on, let's say half a second later, this half a second later. So there's a delay of activation which is getting created. And then the lights, they will create a wave again, but there's no real coupling between the different waves. It's the fact that you have a delay which has been imposed in the switching time of the lights. This mechanism here is called phase waves. And phase waves don't require any coupling, and they can possibly go very fast because it's about the synchronization and the delays that you have. Now, in Drosophila, this is the mechanism which is happening. And in particular, when you go and measure the profiles, you don't see the fronts that are typically expected in the case of trigger waves. You see more profiles that go up in time that correspond to the prediction of the phase waves. And the interesting thing is that I've been telling you that the major determinant between this scenario and this scenario is the time at which the potential is changing. So the speed at which the cell cycle is going. So what you can do is that you can change the speed of the cell cycle by playing on some different factors. In this case is V1. You can play on the cyclin, so you can play on some other molecules, on some other proteins, and slow down the cell cycle. And when you do this, you transition from the one mechanism, which is this one, to the other mechanism, which is the trigger waves that you have here. So you can essentially manipulate by playing onto this molecule and the speed of the cell cycle. You can manipulate the nature of the waves and go from one to the other. And the speed of the waves slows down. This is the wild type, and this slows down by going to the trigger waves. There's some other ways you can manipulate, but I think it's about time that I stop. So let me just tell you, I've been telling you the first couple of hours. There are still, and I think I'm pretty happy of what we understood about these first 2 hours. There's still something that keeps me awake at night. And it's literally true, because while I was preparing the slides, I was thinking about this, and again, I stumbled upon a wall. I don't know how it happens, and it's still a mystery to me. It's the temperature. It's the role of temperature. So I've been telling you the first couple of hours, but the first couple of hours, it's at room temperature. Now, if you cool the temperature in the room where Drosophila develops, in fact, these 2 hours can easily become like 25, 30 hours. Okay? So everything gets slowed down, and this is not just the first 2 hours. These are observations that were made in this paper here, where you see that even later phases that go beyond the 3 hours of gastrulation and so on, you look at them, and you see that they, as a function of temperature, how much they get slowed down. Here, it's represented here. Now, in this paper, they showed that if you rescale the whole duration, the rate at which the whole cycle proceeds is conserved. So it's like there's a clock that is slowed down, and everything proceeds more slowly, but the whole thing proceeds fine, and the rates are kind of conserved, okay? The relative rates. Now, and if you go and fit all this, there's some effective Arrhenius equations that seem to explain this, the activation of these processes with an activation with an effective Arrhenius factor that works. And even this, of course, is very surprising, because there's all sorts of reaction with their proper delta G into the system. So how come there's a single Arrhenius factor that is explaining everything? It's a complete mystery. And even more of a mystery is the fact that you can do some very funny experiments, which was done in this paper here. What you can do is that you create a Y microfluidic system where you put the head of Drosophila at one temperature and the tail of Drosophila at another temperature. And you see what happens. And of course, when you go hot, you go faster, and the tail is going to go slower. So you're going to have more division on the side of the head and less division on the side of the tail. And you can look at what happens, and miraculously what happens is, even if you put something of the order of 7 or 8 degree difference, Drosophila still develops perfectly fine. If you put too much, like 12 degrees, then Drosophila gets stuck. It doesn't develop anymore. But even 7 degrees on 500 microns, you can calculate the gradient. It's something pretty stunning and pretty surprising that Drosophila can kind of manage this. So that's an open thing that I don't know the answer. It seems to be a general property of this regulation in temperature that I don't understand, and it's an open problem. And with this, I conclude. I thank all the different institutions that supported my activity throughout the years, and I thank you so much for your attention.
So thank you for a fascinating talk and a wonderful tour of those exciting moments of development. We have time for some questions.
Yeah, so two related questions, both of which are very large from an outside expert. The first is, well, I know for the reasons you said you were studying Drosophila, do these general considerations, are they expected to translate in development across other organisms, and if so, of which kind? And the second is there an evolutionary pathway to getting to this slightly sophisticated structure that you unveiled?
Okay. So the first question, the answer to the first question is yes. This mechanism, they seem to be quite generic. There's some special features of Drosophila that don't translate to other organisms, because Drosophila, as I was saying, it's a syncytium. So the communication between the different nuclei, it's faster. But I would say from the physical point of view, the mechanisms are very generic. What changes are the molecular players. For example, if you want to communicate and exchange, in the case of Drosophila, you can do by diffusion. In the case of organs that already have cells, what you have to do is to put some receptors on the boundary of the cell and then communicate via different cells. And so the details, of course, will be different. The players will be different, but generically you have at least some of these mechanisms are quite generic. The second question is evolutionary path. There's so it seems to this mechanism seems to have evolved. If you look at different Drosophila, there's some studies that have studied the evolution of Drosophila. It's not possible yet to trace exactly how this mechanism has come up into evolution, but there seems to be a path that leads from very relatively simple things into this more sophisticated stuff. But we don't have a clear view of how this has come evolutionary. We are not at that stage yet.
Other questions? Mike?
You haven't been here since 2016.
Yeah.
Okay, great. Yeah. Thank you, David. This is kind of a specialist question, sorry. The 1 over r squared that you assume,
Yes.
There must be some modification of that with dynamic instability of microtubules.
Yes.
Does that make much of an effect, or is there these are slowly disassembling guys? Or?
There's assembly and disassembling, they get unstable, they get thin, but when you go and you put a process where you put the instabilities, you put everything in, and you measure more or less how it scales, it still scales like 1 over r squared. But yes, we took the instabilities into account. We did the more sophisticated model where they grow, then you have the instabilities, those they shrink. But overall, when they grow, also because these are growing, they are in the growing phase. So overall, there's an average tendency to grow rather than go unstable because the cell cycle is going up. So first they grow up, then they disassemble. So there are some instabilities, but overall there's a trend. And therefore, these instabilities, they don't really matter that much for the 1 over r squared. But there are instabilities, for sure, yes.
So in the last bit, you were talking about response to temperature.
Yes.
I mean, so are you viewing that as there's things special about temperature per se, or this is just an example of like many things, or potentially one of many other things that could invoke similar responses? So different diets of the mom and all sorts of other things.
There's all sorts of things that Drosophila must compensate. Temperature is the most obvious one, and as a physicist, one has a bias toward temperature. But of course, there's many things because the eggs are laid in the fruits, are laid in the organic material. So there's plenty of things that are changing with respect to the environment. Now, the cortex is buffering a little bit because the cortex is rigid and it's buffering a little bit. So temperature is one of the elements that obviously changes, but there's plenty of other things. What strikes me in temperature is one thing, it's also that there seems to be compensation for temperature in all sorts of organisms, including bacteria. They have to compensate for different temperatures. So this compensation to temperature, the circadian clock that some people have worked on, you must compensate the circadian clock with respect to temperature. So there seems to be an overall theme in compensating for different factors. And temperature is clearly one of them, and it seems to be across a number of animals. So that's why I focused onto that with the limited time. But you're right, I mean, there's plenty of other things that you have to compensate for, yes.
I'm wondering, I want to ask something following up on Andy's question.
Yes.
If you look at other insects, is it similar, identical?
It is similar. There's some other insects that have waves as well. So the wave structure seems to be similar. The flows haven't been studied yet. in many insects. The difficulty, I mean, this was the first time where you really could look inside, because looking inside is very complicated for the microscope. You really need to stain something which is very visible, and the red fluorescent protein that they use this. So the flows, it's the first time it's been seen. The waves have been seen in other insects as well, including bigger insects where they've been seen. The microtubules, I think they've been seen in other Drosophila, but not in other insects. So I think it's quite generic, but not all the details are quite the same.
And if I could ask one more related to the waves, perhaps you said it, what initiates the wave?
So what initiates the wave is the fact that the cyclins. So the cyclins are cofactors of the cell cycle. They ramp up during the cell cycle. At the beginning, there's few cyclins, and then they go up in time. So in the language of physics and the potential, what is going on is that you have one of the minima. It's like if you were cooling the temperature into a liquid. So imagine you're cooling the temperature. The cyclins are playing the role of the temperature, and you're making the two minima get closer and closer like this. So there's a moment that you can either jump from one to the other. So the cyclins are playing the role of the supercooling of the liquid into ice. And the question that in physics doesn't get asked, you typically do this slowly, and Drosophila do it fast. And that's the reason why we had to revise these physical models of the bistable potential and the supercooled liquids and take into account the speed at which this is getting done. But the factor is the cyclins that play this role of changing the depth of the two minima.
So, how much did you have to? Is this on? How much did you have to change the timescales to get it to go from the sweep to the trigger? So, in this manipulation, did you have to slow it down by like a factor of 2 to all of a sudden see these trigger waves? Or
yes. So the cell cycle, I think it got slowed down by a factor 2.5, 3.
Okay.
And that allows you to go from one to the other. And then, of course, in the middle, you get something which is a little bit of a mixture of